Good morning, folks. This is Josh Hallman with Vermont Forest Parks and Recreation. Want to welcome you to the third day of the Forest Health webinar series. And I'm going to wait a couple more minutes here, I think, just to let some folks roll in here. But to give you a background on what's going to happen today, we've got a couple really excellent talks lined up for you. We have one on beech leaf disease from Cameron McIntire with the Forest Service. And following his talk, Keena is going to talk to us about spotted lanternfly, and both these are going to be excellent. Some information that we've been looking for, especially here in Vermont. So definitely looking forward to both of these talks today. I want to give a little bit of housekeeping for this talk and for past talks. Let's maybe clean up last week a little bit. If you did attend last week and you're waiting on a credit certificate, that should be coming either today or tomorrow. Apologies for the holdup on that, but they are in process, and you will get those certificates if you're looking for those and waiting for those in your inbox. So rest assured, those will be coming your way. Today, this webinar is eligible for one SAF or CFE credit. It is available for an ISA credit as well, and one Vermont pesticide applicators credit. You may recall with the pesticide credits that at the very end of the talks today, you'll need to answer a survey and answer a few questions about the presentations today in order to get that credit. But I think it's only 3 questions, so it shouldn't be too burdensome. Another thing about that survey in particular, I think some folks last week had an issue with when they closed out of the app, they didn't receive the automatic survey pop-up. If that happens to you today and you do not see the survey when you close out of the webinar, there will be an email that will be sent to you in the next day or two with a link to that survey so that you can fill that out and make sure that you get your credits. So if you do not see the pop-up right after the webinars, rest assured you'll get an email that will still allow you to get your credits. As far as questions go today, it'll be just like all the other webinars we've hosted this way. There is a questions tab in on the right side of your screen there. Please put your questions in there, and I will ask the presenters those questions at the very end of both webinars. So Cameron will present, Melody will present, and then we'll do questions after that. So please feel free to ask those questions via text, or excuse me, via typing into that box there. So that would be great. That's a little different than the chat box that you see there, which is more for communication, but not necessarily questions for the presenters. So that's the distinction. I think that is the housekeeping for right now. If something else comes up, I will mention it at the very end of the talks, but please just enjoy these talks and put your questions in the questions box. And next, I will pass this over to Cameron, who's going to be talking to us about beech leaf disease. So thanks, Cameron, and take it away.
All right, yeah, thank you, Josh, for putting this together. So I'm gonna jump right into beech leaf disease here. And let's see if I can advance the slide. Okay, great. So let's talk about beech. So Josh tells me that most of the folks on this call are coming from a pretty strong forestry practitioner background, so I'm not going to belabor you too much with background info on our host species, other than to say that here in the eastern U.S., American beech is a major component of our mixed hardwood forests, often found associated with trees like maple, birch, and hemlock. It's highly shade tolerant, almost as much so as hemlock. And so this tends to persist in the understory for long periods of time and can take advantage of canopy gaps to trigger a release into the overstory. And as such, it can be quite long-lived. Our oldest known specimens are clocking in somewhere over 200 years old. And I found this great Forest Service technical note from the 1970s. This is at a time when there was a much higher focus on wood products. And within this report, beech is touted as among the big 3 of hardwood lumber species, up there with maple and oak. And to its credit, beech does produce a fine wood product. It's used in all sorts of applications like flooring and fine veneer carpentry. But it's somewhat gone out of favor as a major lumber species in this country. And here we go. So despite its decline in forest products, beech is still regarded as an important species in the landscape due to its value towards wildlife, right? So beech is what we refer to as a hard mass species. And so this produces large crops of nuts on an interval somewhere between 2 and 8 years. trees are generally reproductive past 40 years old, and the nut crops are commonly browsed by many mammal species like deer and black bear. So it has a lot of value on the landscape for that alone. And you can't really talk about diseases of beech without talking about beech bark disease, right? So this has been an important disease in the region for the past century. Here in New England, we're regarded as what's termed the aftermath zone. And all that means is that this disease has moved through over the past several decades, and now you'd be pretty hard pressed to find a beech stand anywhere in the region that doesn't exhibit cankers from this disease. And so this is a complex disease. It's an interaction between a scale insect and two species of Neonetria fungi. And these affect the stem. It results in these unsightly cankers like the ones that you see here in these photos to various degrees of severity. And this largely ruins the quality of the wood, and it can lead to tree mortality in extreme cases, especially when coupled with other stressors like drought. So that's all I want to say about that, really. Let's get down to business with this new disease, which for simplicity, we're just referring to beech leaf disease. So it should be fairly easy to remember. I'm going to start with some of the symptomology so we can all know what to look for when we're out in the field this year. In these photos, I'm highlighting what we term the mild and most common symptoms. And what we've been referring to this to is banding as shorthand. And you can see here that what we have are infected leaves that are largely of normal size, but you can see that there's this dark band that occurs between the leaf veins. And the number of these bands can be variable, but they typically cover less than 2/3 of the total leaf surface. And as you can see from a couple of these photos, it's not uncommon to have a single branch that has some leaves that are banded and then some leaves that are completely healthy or asymptomatic. So this can be pretty spotty, and it does tend to affect different parts of the crown. You actually do tend to see this more in the lower crown, which you know, we can talk more about the advantages and disadvantages of that, but one of the advantages is that it does make it a little bit easier to spot when we're out in the field. And then the second, more severe symptomology is what we refer to as this generalized leaf crinkling. These leaves tend to be smaller than the normal size leaf, and they express these curled and shriveled edges along the leaf margin. And they also become actually like quite thickened and somewhat leathery in texture. And these symptoms tend to express over the majority of the leaf, so greater than 2/3 of the leaf area. And you can see, you know, in some of the more dire examples here, that photo in the center, we have a whole branch which is largely crinkled, so it's hard to imagine that those leaves are providing much function to the tree at all at that stage. And then there's been some really good recent work tracking the phenology of this disease, which has shown that symptoms actually don't really progress over the course of a single growing season. And all that is to say is that when a leaf expands at bud break, the symptoms don't tend to get worse. If it comes out with 5 bands on it, then those 5 bands persist and don't increase later in the year. And we also don't see banded leaves becoming crinkled leaves later in the season. The one exception to this among leaves showing the crinkled symptomology. So in some cases, these crinkled leaves will actually become chlorotic and drop off. And so there's some actual evidence to show that these aborted leaves can reflush, but we really don't know very much about the conditions that have to be present for this to happen. And it's certainly not a given that if it falls off, it's going to reflush. And we do see the presentation of symptoms tend to stabilize over multiple years of infection, usually on the order of 2 to 3 years post initial infection. And here you can see some results of some of the long-term monitoring that's been going on around Ohio for the last 6 or 7 years. And this is showing the proportion of both banded leaves and crinkled leaves. We also have asymptomatic leaves up there as well, just to look at disease over time. But what you'll notice is, first of all, there's a heck of a lot of variation between trees. But what we do see is that infected individuals tend to stabilize around 25 to 30% of proportion of the total crown infected on average. And so this is actually quite interesting. It does seem like it takes about 3 years for the disease to really reach its climax, but it does most definitely persist beyond that. And this is actually most evident when we look at things like leaf cover and dead branches within the canopy. And so I just mentioned that symptoms don't really change too much within a single growing season, but over several growing seasons, we do see significant deterioration of the crown health. And so what we observe is actually, you know, the proportion of total leaf cover declines significantly. You can see, especially on that figure on the left, year 4, 5, and 6, we're talking about less than 20% of our original canopy cover is remaining. And then, of course, proportional to this is the number of dead fine branches that you'll observe. So as canopy cover goes down, of course, we see an increase in the total amount of dead branches. And so this isn't a particularly rapid disease. This isn't like a pest like emerald ash borer and it kills the tree within a matter of a growing season or two. This is something that can persist for several years before causing severe declines within these trees. But it is important to mention that mortality does occur, especially with more of these chronically infected individuals. And so you might have been wondering up to this point what the heck causes this disease, and we can now say with a fairly high degree of confidence that we can attribute it to this parasitic nematode. And the species is called Lidolinchus crenate McIntire, and this is actually a subspecies of a Japanese nematode, though I will say it's unclear at this point if this was actually introduced from Japan or somewhere else. This is interesting work that we're still really curious to find the answer to. And I'll just mention that nematodes do tend to be dimorphic, just meaning that they exist in distinct male and female pairs that sexually reproduce. And here you can see, we have a male on the top left and then the corresponding female on the right. We can see what some female eggs look like on the bottom there, and then the juvenile is just like a somewhat smaller, more indistinct version of this. And these next set of images here, these actually are viewed under just with the naked eye or dissecting scope, and this is approximately what it would look like in the field if we were to break open some leaves or buds. You can see, just barely with the naked eye, they're really these small kind of thread-like worms, and they're also like somewhat translucent. So if you get in there with a hand lens and start breaking stuff apart on a tree that you see some of these symptoms on, I wouldn't be surprised if you were able to tease some of these guys out. And as far as biology is concerned, I'm just going to give like a one-side crash course on nematology because I feel like it's not the forest pest that we commonly think about when we talk about forest health. And despite a lot of shorthand referring to these as worms, like I just did a minute ago, they do in fact exist in their own distinct clade apart from your traditional, what we refer to annelids, which are your earthworms, your leeches, things in that nature. And in fact, they're actually like closely related to arthropods. So these are like what we regard as our traditional insects, right? So these include forest pets like our wood boring insects, their phloem feeders, or ticks, things of that nature. And they're thought to have split from insects around 500 million years ago, where you can see that our traditional worms are actually much older than that. And then just some really interesting things to understand to put nematode in the context of the animal kingdom is that they can both be parasitic like the nematodes we have in beech leaf disease, but many are also free living. There's estimated over 1 million species globally, though only a tiny fraction of those have actually been described yet. And this is the really remarkable thing, is that they are thought to basically compose 80% of all individual animals on Earth. And so conservative estimates put that at about 60 billion nematodes for every one human that's walking the planet today. So there's just like a ridiculous amount of nematode biomass that exists, but they're so small and cryptic that people tend to overlook them a lot of times. And by and large, 90% of all of these nematodes inhabit the top 10 centimeters of topsoil, but they live in absolutely everything, plants, animals, and that includes humans as well. So that they are quite prevalent. They're around you everywhere. There's probably nematodes in your, crawling on your desk right now. And so briefly, I just want to talk about what we know in terms of some of the mechanistic impacts that this pest has on these trees. And here you can see we have two micrographs of the leaf surface. The left shows what we regard as a healthy leaf, and we can see a diseased leaf here on the right. And these red arrows are actually pointing to the stomates. And this is, of course, where gas exchange occurs with the leaf surface. And this is what are basically the structures used to regulate photosynthesis. So very important, critical component of a plant, right? And on the photo on the left, you can see what we call normal function. Stomates are round, they're open. And for the diseased leaf on the right, it's much harder to make out any of these stomates. They're extremely deformed. And in some cases, a lot of them are assumed to have probably lost function. And then the slender lines that you can see on top of the leaf are in fact nematodes crawling around on the leaf surface. You can see there's 3 or 4 of them in that figure right there. And I really want to drive this home. It's not just the outside of the leaf that's impacted. In fact, most of the action appears to be happening on the inside of these leaves. And so again, I'm showing you a healthy cross section here of a leaf on the left. You can see all the hallmarks of a fairly unremarkable leaf. You have a couple different types of mesophyll on the top and the bottom plane. This is bounded by a cuticle, and you got stomates on the bottom, right? And now let me pull up a photo of an infected leaf, right? So check this one out. This one is this is a colorized image here, and what you can see are the nematodes have been colored in blue. And so clearly it's in a very high density within the leaf. It's absolutely crawling with these. So this is actually a cross section of one of those darkened bands that I showed in some of those pictures earlier. And I mean, we're talking about tens of thousands of nematodes within an individual leaf. So they're very abundant. And it's clear from this photo, just comparing it with a healthy leaf, there's a heck of a lot of dysfunction happening here. The basic structures of the leaf have started to break down, and the pests are feeding on parts of the leaf tissue. So this should give you some idea as to the mechanical havoc that this pest can induce within these infected trees. Okay, so where do we stand with all this? So as of fall 2020, this is our understanding of where this disease exists. So a little background, it was first detected in Lake County, Ohio in 2012. And so this is just around the southern shore of Lake Erie there. It's currently found in 7 states in the United States and also the Canadian province of Ontario, just on the north side of the lake there. And 2020, we actually saw the greatest increase in detections. We had 31 additional county level detections in 2020, as you can see from this graph. I'm sure we're all like absolutely sick at looking at exponential graphs over the past year, but for better or worse, this tends to be how diseases progress. And so we expect this to continue before it starts to level off. And it is also really important to note that we don't have any detection in Vermont, New Hampshire, or Maine yet, and I hope it stays that way. But we're definitely keeping an eye on it because it's right on our doorstep. So just last year, 2020, we got some detections in Massachusetts, and then we really saw it blow up in Connecticut. So it's right on our doorstep. And I also want to talk about the fact that we actually have no idea how this is spread currently. We have some hypotheses, possible vectors that might be moving this around. One is it could simply be windblown rain moving around leaves or invertebrate hosts. We do have some evidence to show that there's an association with spider mites, for example. Birds are also being considered as potential vectors for transportation. The nematodes are actually in their highest densities in the leaf buds. And so one theory is that birds grazing on buds could potentially disperse this nematode over long distances. But we also don't discount the possibility of multiple independent inductions. You know, you can see there's some considerable distance between the county level detections on this map. So that's always a possibility. You know, this could have been brought in on nursery stock or something in that nature. And one thing I do actually want to mention is that I've been focusing on American beech, but this does also infect some of our ornamental varieties, such as European and Oriental beech, which are, of course, more common in some of our urban forestry. And our survey efforts are very much ongoing in collaboration with both state partners and the forestry community. I want to take a second to just pitch this app that was created for the sole purpose of tracking and reporting beech leaf disease. It's called Tree Health Survey, and it'll come up if you give this QR code a scan, or if you just Google it on your phone's app store. And what this app does is it allows users to upload location information and photos of beech trees virtually anywhere where you have cell phone service. So this has been extremely useful for building our regional network, and even asymptomatic information is really valuable data to have. So I'd encourage all of you to check this out and contribute throughout your time in the woods this year. That would be super helpful for us. And I also want to highlight some of the regional efforts that we've got lined up to keep an eye out for this new disease. And so the state of Vermont, in partnership with the Forest Ecosystem Monitoring Cooperative and the Forest Service, are going to be establishing some long-term monitoring network plots within beech-dominated stands later this year. And so this is going to be actually following protocols that have been in use in other states for several years now. And so we're excited to be bringing Vermont into the fold here. And among the goals of this work is going to be to relate site and stand level attributes to the presence and severity of this disease. And we're going to be looking at both this new leaf disease and also the beech bark disease complex, because as it moves into New England, it's going to be really interesting to see how trees that are experiencing both of these diseases respond to those detriments in their health. And briefly, I'll just share a couple of preliminary results from the monitoring efforts that have been going on in Ohio and Ontario for the past few years. And what they found is pretty interesting. It's that, well, the pest of beech varied considerably by tree size class, with beech leaf disease being the most common pest in their region. And you'll see that the bark disease, which is on the bottom panel there, was relatively rare in that part of the country, but it'll certainly be, I think, a different story here in New England. And as far as the total number of pests among trees, one interesting finding was that dominant mature trees tended to have the highest incidence, and that was significantly higher than trees in the sapling size class of the understory. And let's see, last one here, finally focusing on beech leaf disease, we see it does tend to be most severe in the sapling size class, which is quite interesting, while scale severity was actually highest within the dominant and more suppressed trees. So this is the scale being the vector for the canker fungus. And Neonectria, the actual canker, showed no obvious patterns in this part of the country. But again, I think this is going to be something that's much more regionally dependent. And I'm trying to wrap up here, and I just want to finish with a couple slides for folks to point themselves to some more resources that are available on beech leaf disease. This past month, we actually hosted our second International Beech Leaf Disease workshop. For better or worse, this was a virtual format. We had over 300 people from 14 countries in attendance, and this covered a whole bunch of topics. We talked about epidemiology, distribution, and as well as research and control efforts. And so I've got a link here. I'm probably going to spam some of this in the chat once my talk is done. And so people can access this. This is actually over 6 hours of presentations that are all time stamped. And so there's also an agenda that you can reference to look through. And so if there's anything of particular interest, folks might be interested actually in some of the control efforts that I am not going to cover in the talk today, mostly because a lot of the control effort is pretty inconclusive at this point, but I want people to be aware that there is some chemical stuff being done that can certainly be applied on maybe not a landscape scale, but will be useful for an urban or small woodlot setting. And then we also have started to gather a lot of great resources that are now in print. One of the great repositories for this is actually through the Cleveland Metro Parks website. They've been an amazing partner. They were actually the ones that first alerted us and discovered this disease in Ohio. And so they have an awesome repository of all sorts of stuff on their website that I would encourage you to check out. In addition to that, the Forest Service just published their pest alert last month on beech leaf disease. And within the last 2 to 3 years, there's been a lot of great peer review research papers that have come out. And so if you want a print copy of any of these materials, or if you get paywalled by any of the research stuff, just shoot me a message and I can easily get those to you. I'm happy to help scavenge those. And that's going to be it for me. I just want to finish thanking this amazing group of partners that have really spearheaded the work around this disease. These folks have contributed to virtually everything from identification, research, the survey and monitoring efforts throughout the US. And it's worth noting that I'm kind of the new kid on the block. All these people on this list have been working with the disease much longer than myself. And so they're really the foundation of their knowledge, and I'm just here to spread the word. So I thank everyone who's tuning in for listening today, and I'm looking forward to Melody's talk and answering questions at the end. So I guess I'll hand it back over to you, Josh.
That sounds great, Cameron. Thanks very much. That was a great presentation. And yeah, I just want to remind folks that if you have questions for Cameron, please put them in the question tab on your the right-hand side of your screen there, and we'll round them up and answer the questions at the end of the presentations today. But next, I'm going to pass it to Melody Keena, who's a research entomologist with the Forest Service, and she's going to be talking to us about spotted lanternfly today. So another pest that's not here in Vermont yet, but knowing what to look for and more about the insect is something that I think we all could benefit from. So I'm going to pass it to Melody now, and take it away, Melody. Thanks very much.
All right, I think you can see my screen now.
Yes,
So
looks great.
this is the adult spotted lanternfly here on this first slide. Whoops, too far. All right, so let's meet the insect. Here on the right-hand side, we have an adult that is showing its red underwings when it got startled. This is one thing that they do. You wouldn't normally see that red except when they're startled. The middle section, this is a newly laid egg mass covered with a waxy coating. And this is one that was laid the same year, but has been roughed up a little bit by adults walking across it. And finally, on this side, this is a maple tree. And you can kind of make out here a whole bunch of adults hanging out on the tree. And as they feed on the tree, they produce a honeydew, and it falls all around the tree. And at the base of these trees, it's going to be kind of black, because a black sooty fungus will grow in the honeydew of this insect. So this insect has 4 instars. That would be the time between molts, starting out with the first instar here that's a little less than about a 1/4 inch or less. That's what's going to hatch, and then they'll molt through these first few instars. They remain this black with white spots, and they're capable of hopping. In the 4th instar is when they start showing the red here with the black and the white markings on it. And then the adult, this is a gravid female. They only show these yellow banding on the abdomen when their abdomen is full of eggs and they're getting close to laying. But normally you wouldn't see the red underwings, you would just see this part. And when the wings are folded over their backs, the adults can be about 1 inch long. So the adults are going to be laying their eggs between September and December in Pennsylvania and that region. They're usually laying early to mid-October on, and then it just depends on the climate, what the time frame is for the egg laying. The eggs here, you can see up in this picture, have slightly different colors, some of them. And here's an egg mass where the waxy cover is off because it's last year's and the eggs are hatched. You can see all these little openings in the eggs. First instars are going to hatch anywhere from April to June, again, depending on the climate, and then they will progress through their instars. And these are some actual photos of a first instar and a 4th instar here. When they get to be adults, it takes them quite a bit of time feeding and maturing before they're even able to mate. And then once they have mated, it takes some more time maturing and feeding before the female is able to lay. And so this is a cluster of some females with some males around them over here in this corner. So where did this insect come from? Spotted lanternfly is native to Asia. It's found in China, Bangladesh, and Vietnam. But it's also become invasive into Japan and South Korea, and more recently into Pennsylvania. It was first found in Pennsylvania in 2014. In South Korea, this is considered an invasive pest, and it has major impacts on their grapes and their peaches. Okay, so this is just a map of where it's at. It was found in 2014 here in Berks County, Pennsylvania, and it has been spready since. The blue is where there are counties that there's a known infestation that's reproducing. And then the red around certain counties are those counties that the state has imposed a internal state quarantine to try to slow the spread of this insect. You can see that some states have no quarantines yet, and other states have quarantines on only part of their counties. You can also see that there are several counties with little purple dots in them. Those are where some stage of the insect has been found, but no infestation has been confirmed. It's also been regulatory incidents. 35 times there have been adults show up in the cargo or the plane coming into California on flights out of Pennsylvania. The green industry also moving plants around. There have been incidences of the insect associated with the plants, New Hampshire, Maine, Massachusetts, Michigan. And the non-ag trade also moving some there. Some of the ones like the ones found in Connecticut, we presume they might have moved on a vehicle. So this insect is getting around both on its own, but also by human-aided transport. This spotted lanternfly makes use of over 70 different plant species, but it strongly prefers the invasive tree of heaven. And this is a picture of the seeds on the tree of heaven, and some incidents here of the insect on it. This is a 2015 map. I'm sure that the tree of heaven, since it's invasive, is found in some other counties at this point. At this point in time, there weren't any showing for Vermont. This insect also is getting into several different crops. This is just a small subsection of the crops it's on. It gets into grapes, and it is killing grapevines in Pennsylvania, has also killed them in Korea. In the fall, when the adults are moving around and looking for things to feed on and lay their eggs on, they do end up in apples and other tree crops, and the nymphs will end up in some row crops. In this case, this is cucumbers. There's been quite a bit of work going on at the Pennsylvania State University in Kelly Hoover and Julie Urban's labs to look at what are the host requirements for its development in the field and what are the effects on hosts. So I'll just briefly go through a few of these. Both in maples and grapes, it has been shown to suppress photosynthesis, so it's actually suppressing the plant's ability to produce its food. It also affects the yield, the winter hardiness, and the sap flow in grapes. It can complete its development on a combination of silver maple, willow, and black walnut in large tree cages with no tree of heaven in the cage. But when you compare it with a cage that also had tree of heaven in it, the development without the tree of heaven was 6 to 8 days, took 6 to 8 days longer, and there were fewer egg masses that resulted. This is important because it was thought originally that if you controlled the tree of heaven that you could stop the movement of this insect, but it has been recently proven that it can complete its development without tree of heaven. And there also been some research done on how the nymphs move through forest blocks, and they can move quite a distance into forest blocks. Again, it depends on the instar, the bigger ones moving further than the smaller ones. There's been a lot of work by several different people on the seasonality or the degree day requirements for this insect. And in each case where I present some information, please watch for the yellow block. That is the base temperature that this particular set of information is based on. In this case, 10 degrees C. So Dennis Calvin at Pennsylvania State University was looking at how long it took at base 10 for the adults to lay eggs. And so he found 1495 to 1710 was the degree day range over which eggs were produced. And then he mapped it out on the landscape. And so I've circled here the section of the map that would contain areas that would have enough degree days. And you can see from this, for Vermont, there are sections here along the New York border and possibly along the New Hampshire border that might have enough degree days for egg development for adults to reach the point at which they could lay eggs. Other research has looked at, Waukee has looked at how to predict where it might be able to do well. What they did in this case is they took all of the points at which they had confirmed that the spotted lanternfly could infest and remain an active population, both in the native range and in invaded ranges, and then use that with the MaxEnt model, which is a specific model that uses this information to predict in the United States where the insect would be able to grow. And these were the traits, the climatic traits, as well as the presence of tree of heaven that were required for maximal infestation possibility. So the first being the mean temperature of the driest quarter of the year between -7 and 7 degrees C, or in Fahrenheit, that's 19 to 45 degrees Fahrenheit. Elevations near 0 degrees above sea level. Degree days here, base 11, greater than 1,000. Isothermality, that looks at how much the temperature changes from day to night versus from January to the summer. And in this case, this insect prefers to have only about 25% of the annual variation in temperature occurring within a day. So it doesn't like to have huge extremes of temperature within one day. But as you can see from the map, most of the known infestations right now are occurring in what is considered the high risk area. So the Connecticut one here, New York one here, and then all down in this region. But there is a possibility, as we looked at in the previous map, of low chance of infestation along both borders in Vermont. This is just another source of information. There are two websites, one that predicts when egg hatch will occur and another that predicts the timing of various stages. Again, this is based on a different base, 8 degrees Celsius. But you can see that it takes a long period of time from adult emergence, this is the adult emergence curve, to actual laying of egg masses here in this case. So I've also been able to actually work with this insect. We have a quarantine facility here in Connecticut, and we've been looking at the effects of temperature both on egg hatch to be able to predict when eggs will hatch, and also the effects on the other life stages, the nymphs, how fast they will develop at different temperatures, how well they survive, and the degree days required for each stage. More recently, an ongoing research looking at the effects of host on developmental time and cold snaps and heat waves. So this has been published. Anytime you see a small black listing, that's the publication that goes with this. So here are the 4 instars. The part in purple is the base temperature. So notice that first and second instars base 13 and 12 degrees C. And I've given you sort of a translation over here. And then the higher instars, 8 and 6 degrees C. The first instars are able to molt between 15 degrees C and 35 degrees C, as well as the second instars, although we're not showing it here on this particular graph. One individual was able to molt at 35. So both of these instars have a fairly wide temperature range, but as they get older, the temperature range narrows. They can complete development at 15 degrees C, although it's not shown here for the thirds, but they're not able to at 35 degrees constant temperature or any higher temperature. So there may be some indications here that this insect won't do well in the far south. And then this is translated into degree days. So for the first 2 instars, similar base temperatures are used, and the third and the fourth, a bit lower temperature for the base. So more degree days required for those instars. And this is a picture of one that's molting. So when they first molt, they look a little bit pinkish. For our first instars, as I showed you earlier, their survival is between 30 degrees here and 35 down here. This is the range here, 30, 25, 20, 15, 35, that they're able to complete development and survive for longer periods of time. Colder temperatures at 5 degrees, a couple weeks, at 10 degrees, about a month, but higher temperatures, less than a week. So host on development. So for first and second instars both, they develop about a similar rate of time on Ailanthus, which is their preferred host, tree of heaven, bittersweet, which is a common viney plant in forests, grapes, roses, and willows. So this is actually the wild rose. So on all of these, they developed about the same amount of time, and these other hosts like basil and maple took a longer period of time to develop, and they also weighed less when they developed, when it took a longer time. Same thing in the second instars. So some hosts they do quite well on, and unfortunately they will develop just as fast on things like grape and rose and bittersweet as they will on their preferred host in these early instars. What we're looking at right now, the cold snaps, we're comparing 15 degrees C with a regime that averages out to 15, but they get 16 hours of 5 degrees C, which is not very good for the first instars in general. But we did find out that they are able to survive and molt at this cold snap regime, but only about 20% of them. The other thing we found out, we were looking at New Jersey collected egg masses versus Pennsylvania collected egg masses, and the time in the first instar at these cold temperatures was quite different between these two strains, which may indicate that as they're moving north, they're adapting to be able to use colder temperatures, especially in these early instars. And heat waves, which is probably not too important to Vermont, but is to the south, at constant 25 compared to giving them periods of 8 hours or 6 hours of higher temperatures, we saw that the mortality increased with the amount of higher temperature that was being applied to them, and also the time that they spent in that instar. So these higher temperatures are really not very good for them. This is a picture of an egg mass hatching. These are the little white insects coming out. They do come out pretty synchronously within an egg mass. And we found that the lower threshold was 7.39 degrees C. They will hatch without temperatures of any other temperature range between 10 and 25 degrees C. But the upper threshold is probably pretty close to 25 because we only had a few new egg masses that were able to hatch at that temperature. We were able to use this then to calculate number of degree days to complete diapause, and then once they completed diapause, to complete post-diapause development before they were able to hatch. And this is a nice microscope picture of them hatching. And as we saw that there were some differences in how New Jersey strain survived the colder temperatures, there's also some indication that they're able to hatch sooner, the populations in New Jersey, which would give them a longer growing period since they are in a cooler part of the region. So there are some indications that as they're moving north, they're already beginning to adapt. In Korea, they have looked at winter egg mortality, which is important to people in Vermont. 100% egg mortality occurs when the mean daily temperature is below 26 degrees Fahrenheit for the months of December through February, or when the January minimum temperature averages 10 degrees Fahrenheit. And so in both of these cases, areas in Korea, they saw 100% egg mortality. So this would mean in the mountain regions, away from the valleys and away from the rivers, you're likely to get 100% egg mortality, even if they were able to lay eggs there. So what's being done to manage spotted lanternfly? As I already mentioned, quarantines imposed by states. We'll look at those a little bit more. Treatments to kill or reduce the spread, monitoring for the infested range, and homeowners' role and options. So we'll go through those quickly so that you can see what's going on. All life stages can hitchhike to new areas, but eggs and adults pose obviously the greatest threat. Here we have an adult on a vehicle. Here's some more nymphs on a vehicle. Here are adults on outdoor items and egg masses on outdoor items and on trees. These are all things that could easily move around by humans. So in the parts of states that are under quarantine, these are the things that are restricted for movement. Landscape, remodeling or construction waste, logs, stumps, and any part of a tree. Firewood of any species, grapevines for any purpose. Nursery stock, packing materials such as pots, crates, pallets, outdoor household items, anything that people might have outside their vehicles, their tractors, their furniture, anything that they keep outside that they might want to move if they move. And the state people work with the businesses to look at the risk of the insect moving along their particular transportation corridor. They train them, they let them get a phytosanitary certificate if they've put things into place to keep the movement from happening, permits that have to be re-upped every year or two, compliance agreements, and verification that they're actually doing what they've said they will do to prevent the spread. In terms of management methods, the initial management method was removal of tree of heaven and setting up trap trees of tree of heaven. and then putting that into management plans to ensure that tree of heaven wasn't available to the insect. So initially what they would do, and all the white top trees, those are the ailanthus, they remove all the female trees, leave a few male trees, 1, 2, 3 here, treat those with pesticide, allow them to attract the insect, and then the insect would die as they feed upon that tree. They're also using various kinds of traps to monitor for its presence. This is the current trap that they feel is the most effective. It's a circle trap that fits around the tree, a mesh that as they crawl up, they get stuck inside of it, and then they end up in this plastic bag. And these big bags are required because the numbers of insects that they're trapping can be quite high and can clog other types of traps. These are pretty cheap to produce and maintain on a yearly basis, and they have greater sensitivity than visual surveys. They have been useful in detecting 2 states when visual surveys were negative. So this is the newest type of trap. This is the old type of trap, a glue trap, but the problem with this is that there's a lot of bycatch or other types of things that are ending up on it, as well as birds and other predatory animals have been getting stuck in it, which is obviously not good. So they're trying to move away from this. What can you do to prevent the spread? First, you need to know what the life stages look like and when they'll likely be present, which is what we covered first. Second, be aware of when you are in an infested area. So if you're driving down into Pennsylvania, be aware that if it's during the time period between April and say November, you might be exposing your vehicle and what you're moving around to this insect. Follow biosecurity best practices. If you go through the region, going to a car wash and giving your car a good wash just before you leave the region that's infested, watching to make sure that they don't end up in your vehicle or on something that you're moving around. And fourth, going the extra mile when you know you're exposed, just being conscious of what's going on. What can the public do? Well, the public can actually scrape egg masses off. If they take and scrape the egg mass off into a container, you can see here as they're scraping, they're actually smashing the eggs, which will kill them. But sometimes eggs will survive the scraping, so they suggest you put them in a container and then add alcohol to it, or freeze it, or dispose of it in a landfill. The public is very concerned about this insect for multiple reasons, but you can get large concentrations of the egg masses and then huge masses of the adult. As we said earlier, the honeydew that they produce is very sticky, and it also ends up producing the black sooty mold. Here they've cleaned this one step, but these other steps at this house are just covered with the honeydew and the black sooty mold, and it can end up on anything and everything. So the public is quite concerned about this insect. So what do we need to do? We have to work together to stop this insect. It's going to take the states, the federal government, the public, and everybody to be aware of it and to help in managing the spread. It's going to require some integrated pest management, controlling ailanthus. Insecticide applications are being used, and more are being developed for different particular uses. They're also looking at using these insecticide applications along things like train corridors, because this insect does move along train corridors. It can move with all kinds of transport. Banding trees, sampling for it, scraping egg masses, looking before you leave. Some important parts of the process, though, are educating the public and businesses about the spotted lanternfly. Some people have thought that the lanternfly will eat their buildings. Not the case. Others have thought that spotted lanternfly will suck their blood or bite people or animals. Not the case. This insect is not going to feed on anything but trees that it recognizes, and licensed professionals that are used for control. There have been people in the public trying to use all kinds of methods, flaming their trees where the insect has laid their eggs, and all kinds of other things. not good, obviously trained professionals are a much better method to use. And being conscious yourself. This is one of my coworkers when we were out collecting spotted lanternfly egg masses. He didn't even know he had a spotted lanternfly right here, and here it is up close. So keeping your eyes open, being aware of the situation. And I will end there.
That's great, Melody. Thanks so much. A lot of great information there and a lot of information that is tailored to us here in Vermont. So I definitely appreciate that. Yeah, we have a few questions that are in the questions box right now. I'd encourage others to continue to ask your questions that way. And I will try and relay these questions to our presenters and get some answers for us here. So Melody, while I've still got you on the line here, one question that came in is you were showing some figures with rose in there. Is wild rose invasive multiflora rose, or is that a different species of rose that you're talking about?
Yes, multiflora rose. That was what we used in our experiments. They love it just as much as they love bittersweet.
Got it. That's great. Thank you. And this is a question that I actually had. In looking at some of those pictures of the egg masses for spotted lanternfly, they share some characteristics with gypsy moth egg masses. I didn't know if there's a easy way to tell the difference between those two, or any advice you have, since we're seeing more gypsy moth egg masses up here these days.
All right, so the gypsy moth are going to be in the tan range from a light tan to possibly almost a dark gray, but usually not anything in the gray region that the spotted lanternfly egg masses are. The spotted lanternfly egg masses are really kind of a light, little bit of pink in the gray, and it's going to be although they call it a waxy covering, it's really kind of a dust once it dries, and it flakes off really easily. Whereas gypsy moth egg masses are covered with the female's hairs from her body. So you can tell that it really looks like a hair or a scale, whereas this is more of a foam that just kind of brushes off easy.
That's great. Thanks for that. Appreciate it. Okay, I'm going to switch the camera here for a few questions. Cameron, the first question we have in the box here is, are trees that already have beech bark disease more prone to contracting beech leaf disease? You mentioned a little bit about survey efforts to kind of determine that, but do we have any preliminary information on it?
That's a great question. There is no evidence yet that would suggest that a tree with the bark disease would be more likely to get the leaf disease, but I think there's a good amount of consensus and evidence from other disease interactions that if you're a tree that has both the leaf disease and the bark disease, you are probably like going to be more predisposed to mortality. Just for the sheer fact of having multiple stressors.
That's great. Thanks, Cameron. Another question, does beech leaf disease stimulate root sprouting like beech bark disease does?
That we don't know. That's a great point, though. Beech does a lot of that vegetative clonal kind of sucker distribution. So one theory is that maybe nematodes can actually take advantage of that and move from tree to tree in that nature. I mean, it would have to go through the vascular system, but again, there's no evidence of that yet. And part of that is just because this is so new, and this is something that's on people's radar, but has not been tested in any systematic way.
Got it. Do you know if there are any natural controls of nematode populations? Specifically the causal factor of beech leaf disease?
That we don't know yet either. There is some work being done in Japan to look into that, because that's where we presume that this subspecies is native to. And so if there was a natural predator, that's where we would expect it to be. We do know that the nematode that affects oriental beech in Japan, it doesn't cause like the severe symptomology that we see here. So there is probably some natural resistance going on in that part of the world that we're not seeing here. But as far as like an insect predator, that hasn't been identified yet.
Got it. Is there any evidence that beech leaf disease is reducing mast production at all?
Another great question. We don't have any concrete evidence of that. So the longest monitoring has been going on since about 2012. I think I mentioned in my talk that masting cycles are something that is really a hotbed for research. Like for all tree species, like so little is known about what actually causes masting events. We know that there is definitely a climate component to it, and that there is a lot of synchrony among populations when it happens. But when it comes down to it, like we don't know a lot of the root causes of masting. You know, at some level, trees will mast when they've built up enough carbohydrate over several years, right? So they've stored up enough sugar in order to flush out a huge crop of nuts. So one would have to assume when you have a disease, especially a foliar disease, that's going to compromise carbon uptake. And you can make a case either way. You know, with some trees, we see almost like this survivalship sort of response, right? Where if a tree is detecting imminent death, it'll just dump all of its stored carbon into like one big hurrah and produce a nut crop. But you could make a case for the opposite. You know, if you're just really starving for carbon, then you just don't have the resources needed to produce a bunch of nuts. And so I think that is something that is going to be useful for the long-term monitoring, because masting isn't something that happens every year, right? And so we're gonna have to look at many years worth of data before I think we can draw any conclusions about that.
That's great. Thanks, Cameron. Melody, a couple other questions came in about spotted lanternfly. The first being, are the circle traps baited, or is being on the host tree enough to attract them?
They're still working on baits that will actually enhance the trap catch. So that's still a work in progress, but the insect tends to crawl up the trees, and that's how they end up in these traps. So they're using their behaviors against them.
Great, thank you. And another question about SLF. You know, is there concern about the prevalence of invasive plants like bittersweet, tree of heaven, and multiflora rose aiding in the survival of SLF as it spreads? And I'd imagine there is some concern about that. And if so, are there any steps that folks, whether federally or at the state level, are taking to mitigate that and to address that?
At this point, no. They've concentrated on the tree of heaven. Obviously, this insect can use so many different hosts, and it does quite well as in the early instars on multiflora rose and bittersweet, and also uses poison ivy and other things that are quite prevalent in the understory. So it would be impossible to completely get rid of its primary hosts for the early instars in particular.
It's unfortunate. Let's see. That looks like all the questions that are in the questions box right now. I'll give it one more minute for folks if there's a last minute question. But I do want to take this opportunity to once again thank Cameron McIntire and Melody Keena for providing great talks today. We really appreciate that. I'd also like to put a plug in for next week. Next week will be the final webinar for this series this year. And it'll be discussing forest bird monitoring in Vermont. So that could be an exciting one that's a little more wildlife based, or shows the intersection of wildlife and forest. So kind of an interesting talk if you're able to tune into that. That one will be recorded as well. And I should have mentioned earlier that this is recorded as well. So if you came in late, or you have somebody that you'd like to tell that they should tune into this, these are being hosted on our Vermont Woods website, and you can just do a Google search for that and it'll pull it up. You'll be able to view those on demand and even receive credits for those. So the person to thank for that and for helping set up all these webinars for this series is Kate Borer. So definitely appreciate her help with that. Again, when you close out of this webinar today, you should have a survey that pops up that includes questions to answer for pesticide credits. If that does not pop up for you, you should be receiving an email in the next day or two so that you can receive those credits after filling out that survey. And for anybody waiting for a certificate from last week, again, I will be sending those out in the next day or two. So stay tuned for those. But at this point, I think there's no more questions that popped in there. So I think we'll call the webinar closed for today. Many thanks for attending, everybody, and for asking some great questions, and hope to see you here next week.